Okay, well, I think we'll just slowly get started then. So good morning or good afternoon, everybody, depending on where you're based. Welcome to the 45th Structure Meets Function webinar. So we have two speakers today. Both of them have accessed Instruct-ERIC services, technologies through the canSERV project for their research. So we have Robert Konrat from the University of Vienna and Maria Macias from the IRB in Barcelona. And our moderator today will be Radovan Fiala. I will hand over to those guys in just a moment. But first, I just want to give an introduction to Instruct-ERIC for anybody who is unfamiliar. So Instruct is an organization with 17 members across Europe and 12 centers providing funded access to structural biology research infrastructure for researchers in those member organizations, as well as access to structural biology technologies. There's also opportunity for training workshops and resources, also a 3 to 6 month internship program. The call for that is open at the moment, so take a look at the Instruct website, and also opportunities for research and development funding as well. In addition to this, Instruct will be hosting the latest biennial structural biology conference. This will be taking place in Brussels, in Belgium, in May. Registration for this is still open, but closes at the end of March on the 31st. So do register and join us in Belgium for the conference. Also on Thursday, the abstract submission deadline closes. So to participate in the poster session or to be selected for oral presentations, the abstract submission closes on Thursday. We also have fellowships funding available for students to attend the conference. This call also closes on Thursday. So do take a look at those and make sure you submit your application as soon as you can. Okay, and there's a QR code to register as well. Okay, but that's all from me, just a quick introduction. If you do have any questions on the conference or any other aspects of Instruct, then please get in touch at support@instruct-eric.org. And what I'll do now is I'll hand over to Radovan to introduce our first speaker. So thank you, Radovan.
Then we can move. Oh, okay, yes, there is a question.
Was a great resolution for the... Exactly. So higher fields are essential for intrinsically disordered proteins because of that problem. And in particular, carbon detection, direct carbon detection, which was considered to be problematic for proteins, is an ideal tool for intrinsically disordered protein. And in this one paper I mentioned, which was a nice collaboration again with the group in Florence, it was shown that even at 1.2 gigahertz, carbon spectroscopy of IDPs can be very, very beneficial because you gain a lot of resolution in an indirect carbon dimension.
Okay, thank you. I do not see any other questions in the chat, nor I see any hands. So in that case, we can move over to the next talk, which will be given by Maria Macias from IRB Barcelona. She was originally an organic chemist, but for the last 20 or perhaps more years, she has been studying mechanisms of diseases by structural characterization of macromolecular assemblies. Specifically, her research involves TGF-beta signaling pathway, and she uses both structural biology methods like NMR, X-ray crystallography, or electron microscopy in combination with biochemical and functional studies. So she will tell us more about her research. Please, Maria.
Hi, thank you. Thank you for giving us this opportunity to present our results. Can you see the presentation is working?
Yes, we see it. It's
Good.
Perfectly fine.
Okay, so the work I'm going to present today is not about the structures, but about finding molecules that modulate the function of the proteins we are working with. And so we applied and got granted this canSERV application, advancing SMAD4 targeting therapeutics. And so we try to find advanced technologies for personalized oncology. And let me see. Yes, so the main aim of our work, we had already identified some hits that interact specifically with SMAD4. And SMAD4 is one of the proteins, I will explain you later, that transmit the signaling that starts at the receptor level in the TGF-beta receptor family and then goes to the nucleus and modifies the gene expression. And this is a very well-conserved signaling cascade, and unfortunately is mutated in many diseases. So the need we found was trying to address this regulated signaling by putting the focus on SMAD proteins, and in particularly in the SMAD4 mutations, because some of these mutations lose the capacity to interact with other SMADs or gain other capacities, and this is directly related to the diseases. And we think this is important to look at this because it's important to have new therapeutic targets, not only to focus on receptors, and also because we hope by targeting the modulators, we might find new ways to treat these diseases with less impact in the quality of life of the patients. And then our final goal is to bridge really the gap between the hit discovery, which is done in many labs, but then we stopped there and we thought we should do something else to move these hits towards something that has a use for our society. So since this is going to cell biology was for us a big jump because as you said before, our expertise is in structural biology. So we found this canSERV a great opportunity for us because there were platforms there with expertise in cell biology and also in studies of toxicity. And we thought it's not just to get the results, what we need is to have also the advice, because we can set up with them some experiments that later on we can translate to our lab, but we really need to know if the results we are getting are meaningful. So I think this is the strength of this application, that is not that you get some results or access to facilities, it's the knowledge you gain when you talk with the people working there. So we asked for two set of experiments. One was the live-death viability and cytotoxicity for our compounds, a few of them that we selected. And we also wanted to know if the results observed in these experiments were related to our proteins. Because the targets we have identified, we identify them as binders of pure proteins. And we want to know if in cells this is also happening or if there are other off-site targets of our compounds. And then the in vitro efficacy and toxicological responses. So this is the team, and as you see, we are several people from our institute, but also many people from the facilities. So we really appreciate the help, but also we are in contact with patient associations because for later on we might need to get samples from biobanks related to them, and we thought it's important that they are involved from the beginning. So this is just a cartoon to let you know with the system we are working. So in general, so what we have here is really a simplification. This is a receptor that is a dimer of dimers and is activated upon binding of the hormone. Actually, the receptor is a kinase. So what it does once it's activated, it phosphorylates some SMADs. They associate with SMAD4. They form quaternary structures, and then those structures go to the nucleus and do the control the gene expression. And so most of the current strategies to modulate the function go to the receptor because it's accessible, and but also it has really severe drawbacks because you block all the signaling cascade. So normally this has many side effects, and most of the compounds or antibodies that they have been developed fail because of this huge amount of side effects that they induce. So we thought, why instead of inhibiting, we try to modulate the association of the SMAD proteins, and then we modulate the function. Perhaps we don't need to inhibit it, but just to reduce or to stabilize some of these interactions might be sufficient and might be efficient, but we might not induce the same kind of side effects. And this is what we did. So we started with open screen libraries. We had access to 100,000 compounds. Out of them, we got 25 stabilizers and 160 destabilizers. And these experiments were done against SMAD4. We could identify them using calorimetry, so DSF. And for those that they were looking nice and that they have a nice saturation curve, we did orthogonal validations as the one shown here with the spectral shift. But then we also thought that perhaps instead of just looking at SMAD4, we could target the quaternary structure of the SMADs. So we set up an HTRF experiment with SMAD4 and SMAD3 as a heterotrimer. And we evaluated the effects of binding 160,000 compounds from a library at the IRB Barcelona. In this case, we got more hits, 2,435, and we group them again as stabilizers and destabilizers. And we categorize them in 16 chemical series. And one of them actually have compounds in common between this library and the previous library of the open screen. So these hits were the ones we used to apply for the canSERV. And this is how we did the hit optimization with the heterotrimer. And as I said before, we have classified the compounds into types. One we call a stabilizer, which are compounds that are bound to the heterotrimer, and they maintain the heterotrimer, and the stability of the system is even increased. But others, they interact with the heterotrimers and they disrupt this interaction to a certain extension. And actually, we think that both systems are useful or could have applications in the future, because one of the problems is that the variants associated with the SMAD proteins are classified into types. Those that cause a loss of function, and others that produce a gain of function. So we thought we could use either the stabilizers for these conditions, or we could use the stabilizers for cancer and fibrosis at the beginning. But then after talking with the facilities, they told us that, well, actually, it is not so relevant if there is a loss or a gain of function if your compounds are bona fide hits, because then we can modulate those hits to either acquire other capacities or even be converted in other kind of molecules like glues or protacs. So we thought this is good because as we have very good binders of both types, we can choose the best ones and move forward. And later on come back and use other compounds that were previously identified that might be similar to the hits. So we classify them, the stabilizers in this case, in all these groups. We choose here some representative molecules of this cluster. What we have here represented is the percentage of inhibition. And here, for instance, you see that we have a lot of compounds that are strong binders, but we choose this one because actually there is a gradient of different affinities already in the cluster. So by comparing the molecules, we know already which groups could interact better and which substituents in the molecule might disrupt the interaction. So we went to the first platform, which is the InnoPharma, and they set up these live dead cell screening results for the project. And so we choose the A549 cell types, which is a model well accepted for modeling metastatic progression, because we were interested in testing our compounds later on in patient-derived cancer cells. So the assay is the control to distinguish between phenotypic inhibition and generalized cell death. And our idea was to verify that the new drugs we have identified effectively targeted the post-EMT cells or mold while sparing healthy epithelial population, so that they don't kill everything, but that they just kill some types of cells. And then we assess the differential survival of the phenotypes when exposed to the compounds and check the viability. I show you some figures. This is the protocol they established for us. And so here we have how these cells acquire the fibrotic phenotype they observe after the incubation with bleomycin. And then we also here have how the presence of two of our compounds affect the cells. So these are the cells in the ideal state. Here is when we induce death. For comparison, one of our hits is also killing the cells mostly. The other one behaves slightly better. So what they did was to classify all our molecules, the ones they tested, and choose the 8 best ones. And well, here we have how the representation of all hits they test and the 10 bests. And here we have a list of the IC50s and Emacs that we obtained for these compounds. And you see a couple of them are very good. Several of them were actually quite good. And we choose these two for the second platform for testing the ATME properties. But before that, we also request to test a correlation between our compounds identified in vitro as binders of our purified proteins. And we wanted to correlate if those effects were also visible in cells, and if we could follow how SMAD4 changes in the presence of the compounds we have identified. At the beginning, we thought, in our proposal, we thought that we could transfect our protein and then test the effect of the compounds in this transfected protein. But after discussing with the platform, they told us that perhaps makes much more sense if instead of doing this with a transfected protein, we follow endogenous protein. And for that, we need to modify the A549 cell line with some CRISPR and Cas knock-in. And what we chose was to introduce the hybrid peptide sequence in frame upstream of the SMAD4 gene to detect this protein either with antibodies or with a reaction with a large bit protein. Okay. So the idea was to see the interaction to be able to measure the protein destabilization and to have like a luminescent readout that we can follow SMAD4 degradation, for instance, or accumulation in the nucleus or in the cytoplasm. This is how we did the modification, and this is how this cell line looks like. It looks pretty much like the unmodified cell line, so only the modification in SMAD4 is present and is not affecting the behavior or the phenotype of the cells. And the platform is now, they are now testing the distribution of SMAD4 in the presence of the compounds. And so for the second platform, which is the SynTEF, in Trondheim, we just provide them with 2 of the compounds that were the best ones behaving in terms of the cell system we have used, and they are now trying to test if those compounds behave well or bad in some of the properties they are exploring. And it's important to do this right now because now we are choosing the scaffold and we are also choosing the modifications we are following. So now is the time to say, well, these ones are very toxic. To get them not toxic, you will have to do a lot of chemistry, whereas these ones are good already. So it's just to improve them. And so I think it's very useful to do this right now and not to wait until having a more developed and advanced compounds. And in parallel with this, as we wanted to know the effects of the variants, the SMAD4 variants in binding to the compounds, we got a second canSERV grant to test the biophysical analysis of those variants in SMAD4 in cancer. And the synergy with the other project is that we want to focus on those patients in the future, those patients that will benefit the most from our compounds. So what we want to know is that the compounds are able to recognize these variants present in those tumors, because some of the variants affect slightly the structure, some others, they really damage the structure. But for the ones that they affect the structure a little bit, they also affect how the SMAD4 interacts with other SMADs. And we want to know that in advance because depending on how the protein associates with other partners, we might be able, or we can select some compounds that we have that do better stabilization, for instance, or that behave better in a given set of conditions. So what we are studying, thanks of this second canSERV project, is the stability followed by nano-DSF, also the quaternary structure dynamics. So we were using several techniques that tell us the percentages of the complexes that are still alive in the presence of the variants. We do see that for those described as the gain of function, we do see an increase in the amount of stable of complexes, and they are also stable for longer time than other variants that the affinity is affected and decreased. Okay. So we are also identifying different stoichiometries and population distributions of the complexes, which were unexpected because at the beginning we were working with the idea that most of these complexes are heterotrimers, but depending on the variants, we observe other kind of associations. And we wanted to quantify all these dissociation rates, not all, a few that we have selected as examples, just to have an idea if some of the effects we observe in the complexes are not due to the presence of the complex itself, but the dynamic of the formation of the complex, if the disassembling of the interactions are faster or slower. Okay. So the idea is that use to find like a mutation-specific compound selection, and then try to reduce to the maximum the side effects of using compounds in patients that that particular compound is useless because the mutation they have is not actually forming the complex we thought. So those molecules will not help these patients at all. We also did some to identify 2 nanobodies, because the nanobodies can also help us to monitor how the SMADs associate. We found 2, and one of them prevents the formation of the complexes, and the other one is able to bind whereas the complexes form. So we have tools that tell us as controls, now I can add this nanobody and reduce the complex, the amount of total complex I have. Or if I use the other one, I can follow the movement from the cytoplasm to the nucleus of our protein of interest. And the good thing of this is that we can label these nanobodies, and they are not expensive. So can be used in high throughput analysis if we need to. And it's not that you need a huge inversion like with commercial antibodies. And we are also exploring the structures of those complexes, of the SMAD complexes in the presence of the nanobody using cryo-EM, just to have a platform. We got really good resolution, as you can see. And we would like to have a platform to explore those compounds that bind to the complex and stabilize the complex further where they bind. Okay. So as a summary of our project, I will draw here that, well, now we are working with cell lines, as you see, but our idea is to optimize the conditions that we can start using patient-derived cell lines that are available in biobanks. We have also the structures with the hope that we can map the interaction site of the molecules and the nanobodies as tools. And our hope is that among these groups of molecules, we can find out 1 or 2 of the stabilizers and another one of the stabilizers that are really useful. And for that, I have here the description of how we envision the continuation of the project. So together with the group in Galicia, we got granted a grant here for la Fundación La Caixa, that will allow us to continue the identification and the development from the hit we have to the lead optimization of the compounds. And our idea is to focus in the disease-specific validation, using to specialized cellular models, and to move to in vivo proof of concepts, of course. And then the other thing is that during the time after the conversations with both platforms, they encourage us to start doing all the paperwork you need for the ethical approval of working with patient cell lines and so on. And we have started and got some of the approvals already. So this means that we can continue with the project and hopefully move it to a preclinical phase, not too far in the future. And with this, just I want to finish. This is our lab, and thank you to all the collaborations and to the funding agencies that have helped us to move from just an idea that why we don't look for complexes with compounds to the point where we are now. So, and thank you for your attention.
Okay, I thank you, Maria, for your presentation. And now there is a time for questions. Do we have any questions? Either from you either raise your hand or write them again in the chat. In between, I may ask how many of these lead compounds you expect to reach the preclinical phase?
How many? How
Yes.
many? we don't know. We have now 16 clusters, and some of the compounds are really, they bind actually already in the nanomolar affinity. So we think some of them look very promising. I don't know, it's hard to give you a number, but we think perhaps 6 could go to advanced.
Yeah, my question was very basic because I know it's a limited. First, there are many limitations for for application in vivo, and second, that's these studies are difficult. So my question was whether majority of them or just a few of them. So it looks like it's a very selective, very tight selection that goes further.
Yes, because as soon as we start working with mouse models or things like this, we will have to reduce down to, as I said, perhaps 6 compounds, no more. It will depend very much if we can use models that they are already developed, because for cancer research, there are many models already available. So one of the possibilities we have is to contact groups that they already have developed these models. If they are interested in testing the compounds, it will be very, very helpful for all. And also the same applies with cell lines. Sometimes you think, I have to do everything, but we don't need to do everything, because there are other researchers that perhaps with a different purpose, they just develop something that is useful. So I think one of the powers of the times we are living now is the communication with other groups. That perhaps before was not so easy, but now you get immediately access to other potential researchers and collaborators. So I think I'm optimist in the sense that perhaps we can get more results, not only those that we can set up in our lab or with the platforms we have access to, but others that because we spread the word and they offer to help us.
Okay, so good luck with that.
Yes.
So are there any questions for Maria? I do not see any hands raised. Let me check the chat. No, there are no further questions. So I think we can wrap up. So with that, I give the word to John for final remarks, if any.
Thank you, Radovan. Yeah, that's great. Thank you, everyone. And thanks to our speakers as well. And we will see you all at the next webinar in April. And we will hopefully see you all in May at the biennial conference too. So thank you very much, everyone.
Okay, bye-bye, everyone.
Bye-bye.